All right, 12:34, let's get started. So hi, everybody, and welcome to the Data Sciences for Biomedical Professionals workshop series. I'm Dr. Brown, and I'm excited to be here and talking about these different aspects of data. So just a couple of things to kind of note that we have going on right now. We got an email yesterday about a pre-workshop survey. If you all are interested in taking this survey, it's a part of a research project that myself and Dan, who I'll introduce in a few minutes, are working on to better create learning resources for individuals in the biomedical space for learning to work better with data and using a variety of automation programs like R and Python. So if you are interested into that, the link is also in the chat for you to take that pre-survey. We will also be recording this session so that it can be uploaded in the future. If you have any issues with this, please let us know. And if you would like to remain more anonymous, you're more than welcome to chat us individually in the chat feature so that you're not using voice or doing anything you're not comfortable with. So with that being said, does anyone have any issues with the recording? Okay, I don't see anything, so I'm going to start recording and get rolling. So I'm going to tell you all a little bit about myself. So I'm Dr. Brown. I am a computational biochemist by training, but a data enthusiast who uses data science in a variety of projects I work with here at Virginia Tech. I'm an assistant professor and a science informatics consultant in university libraries. So I work with a lot of different people all over campus to integrate bioinformatics and molecular modeling, as well as data science techniques into research projects, while also running my own lab that looks into modeling of different disease states and of different kinds of ways to engage undergraduates in research experiences. I do a little bit with health and data analytics, which is why we're here today. And I'm very big for working with data reuse and reproducibility and being as transparent with methods as possible. And I also have affiliation in the Department of Biochem and in the Academy of Data Sciences. How this workshop is going to work today is that you're going to have your main instructor, which is going to be Dan Chen, and he'll introduce himself in a minute. And I'm going to be here for helping, but we also have two helpers, Jonathan Berganti, who's a lecturer and the data bridge manager in university libraries, and Carter Gottschalk, a research assistant who will be taking notes and then able to facilitate and help people one-on-one if you're having any problems in breakout rooms that we'll put you in as we go along. So we're excited to do this in a very collaborative way, and it's one of the best ways to learn how to use different coding languages as you work with your data. All right, and now I'm going to turn it over to Dan, who is really the creator of a lot of these resources and these workshop series in general. Dan is going to talk about himself, and then we'll get rolling from there about how everything's going to go.
Okay, hello. All right, so I'm Daniel. I am a PhD student here at Virginia Tech in the Genetics, Bioinformatics, and Computational Biology program. You can consider myself as a data science educator. I've been involved with the Carpentries as an instructor and currently is their community maintainer lead. And last year I was one of the Rstudio interns where I worked on using R to help create lesson plans and creating an auto grader system so you can submit code to get checked. And the names of those packages is Grade This and Learn R. And part of today's workshop is related to my dissertation, which is studying data science education and pedagogy for the biomedical sciences. All of the materials that we have presented are online, and so you can go to the website, which is also, it should be like pinned at the bottom of my video. And the slides are also available if you want an actual picture of the slides. Just so you know, the slides are literally screenshots from the lesson materials. So there's really nothing, the slides just make it easier to present things during the workshop. Carter is the head of or in charge of taking notes on the Etherpad. So there is also a link to the Etherpad at the bottom of my video. And what that allows us to do is we are able to take collaborative notes. All of the links that I have provided will be pasted up there. So if you really need like one place to bookmark for today and Thursday, then that would be the link to go. It is a regular notes file, and then we'll save that off into another location, so it'll persist after the workshop. And then last but not least, if you are caught up in any of the technical jargon or technical terms, you are free to look at this project that is maintained by the Carpentries, which is a glossary of technical terms. And so you can use that as sort of your quick dictionary as we go through the workshop. So a few logistics. First, if you don't see my presentation screen, I'm not actually presenting off of a screen share in Zoom. So what you should be able to do is find me in the whole gallery of people in Zoom, click on the little 3 buttons, and then pin my video. So if someone else takes the mic, it doesn't actually pull away from my current screen. But it seems like everyone is muted, so that shouldn't really be the case, and everyone should be able to see what I'm presenting. As far as your actual screen layout for the class, think of your current computer screen. If you are, this really applies if you only have a single monitor to work with. Think of your current screen as 4 quadrants, where you have just the Zoom video as 1 quadrant. Rstudio, which we will be working on in the next hour, will be another quadrant. And then you have your Etherpad open in one, and then the participants and Zoom participants and chat in another. And you can resize them vertically as needed. You don't really need that much space for the participants panel and the Zoom chat. The main reason for the participants chat is having access to the yes, no, those little buttons. So if everything's okay so far, in the sense that you can see my presentation screen, most of the things are legible, can you use the participants panel to click the green yes, and then we'll know that people are sort of, things are working. And if you have an issue, hit no. At any point during the workshop, if you have a problem that you don't want to bring up in chat, or you're having like, my computer is totally borked, click the no X, and then one of the helpers will be able to help you figure that out.
All right, and for everyone just joining, we're excited for you all to be here. We've got a couple links that where we'll repost in the chat right now for you to get acquainted with things and have access to our resources with our lesson materials, slides, and a collaborative note space. With that, though, we'll kind of get rolling from there. Does that sound good to everybody? All right.
Yeah, and the main takeaway is the 2 links that are pinned at the bottom of my presentation video. Those, if you ever get lost, just go to either the Etherpad or the main materials page, and that should take you to any of the links that you may have missed during in the actual Zoom chat. So this series of workshops is actually part of a research study, and from the overall findings that we got, where people are sort of indifferent about using a programming language, working with their data. They are not really programming in general while they're doing their analysis work, which sort of makes sense why they're sort of indifferent towards it. But they have this strong belief that they need access to their original raw data set to be able to do a repeatable analysis. And so this is sort of the gaps that we're trying to fill in this series of workshops. This is really just the first set of introducing us into the world of programming for data analysis. And so we're trying to bridge that gap between you wanting to work with your raw data set, but you either don't want to or don't have the skills, or you're trying to learn all of the skills for programming off of that raw data set. And the study actually created a set of personas that you can read more about. It's on the actual main workshop page. And so as our first exercise, just so we can sort of deal with any problems right now, but also for people in the chat to sort of know each other, go to the personas page and sort of talk amongst yourselves with which actual persona do you most identify with? And I believe breakout rooms were set up for this. If not, eventually they will.
We have breakout rooms, so you guys can take a look at who you most identify with for personas and go to the breakout rooms. We'll go there for about 3 or 4 minutes and then pull you guys back in as we keep moving forward. Look at me go. I made breakout rooms.
Oh yeah, I was gonna say, I thought I don't see the breakout room, so I don't know how they're made. Were they just 4 rooms that were those 4? Okay.
There's 3.
Originally,
I put 1 and 5 in one of them because I couldn't do the math fast enough.
oh, but so it's are they all random or were they labeled? Oh, okay, cool. Okay, that's what because when you described it, I thought I was like, wait, did you label them and then they're supposed to self-assign, or are they just gonna talk amongst themselves?
Hey, the notes thing, it says I was typing too many messages and it's limiting me and disconnected me. I don't know what to do with that.
Really? On Etherpad? It rate limited you?
Yeah, and for some reason when I'm typing it's coming up in like pink highlight.
Yeah, that makes sense.
Okay. Yeah, it did. It said you were limited and then it disconnected me. I refreshed, so it was fine, but just note if there's like a delay in what I'm typing on the notes, that's why.
Oh, that's interesting. Yeah, so this isn't so I was trying to originally make the Etherpad through the carpentries page, but then there's a very big blurb at the top that says don't use this for your own work. So I was like, oh dang, that was unexpected. So that's why it says Wikimedia, because it's like that's the more public one, and I guess they have a stronger rate limit to it. And then for the next time, which is probably not going to be on Thursday, we'll see how bad or good this is doable. If it makes it easier for you, you can put in a text editor and copy paste things over if you get super rate limited.
Okay.
But I might try to figure something out. Actually, I don't know if I have time. It's not going to happen by Thursday, but for next time, I might just set up my own Etherpad server, and then we can deal with it that way.
Okay.
Is it your actual? Oh, actually, do we have a timer going on?
Yeah, about 1 more minute.
Okay. Yeah, we'll see. Just let me know how badly you get rate limited.
Okay. When they come back, do you want to ask if anyone wants to put in the chat what their persona is or talk about it?
Yes, I will just ask them to. Give me a second.
All right, closing the rooms.
Okay, so welcome back. So if you would like,
2 more rooms waiting to come out.
No.
All right, there we go.
Okay, so welcome back, everyone. If you would like, just type in the name of the persona that you mostly identified with. It's actually kind of interesting to see who's actually attending the workshops. Just a little bit more background about the personas. They are really just fictional characters that are representative of of a audience. And so it sort of helps direct and pinpoint like the actual content that gets created for the series of workshops. All right, so between today and Thursday, we will start off with learning about some of the basics of spreadsheets and when they are useful and how to use them more effectively. And with regards to the personas, yes, there's definitely a bunch of overlap between them. And so, yeah, they're not any one person's going to be completely relatable to only one, but it sort of gets a better sense of the group as a whole. Okay, so between today and Thursday, we're going to start our learning process. And pretty much for most of the people that in our original set of surveys where we created personas, there was a large group of people who actually didn't know how to program. And so you'll either be somewhere towards the beginning or somewhere around this, this first slope. And the point is that, yes, there is going to be a learning curve, and after a certain point, you are going to realize that, like, okay, I've know enough to start doing my own work, and then you start realizing that there's actually a whole other world of stuff that you don't know. But there's communities around R and programming and in the biomedical sciences to help you all learn this stuff. And even me today, I sort of am on this very far end where I actually just know what I don't know and I'm okay with it. But with experience, you are able to pick up and learn that other new skill without worrying about like the actual like syntax of the R language. So it really does take time, and just know that there is a community to help you progress and learn when you get stuck. And so, speaking about getting stuck, the debugging process, this is something that everyone, myself included, goes through. And one of the things I usually tell my students is she always asks, like, how do you know how to do this stuff so quickly? And it's literally because I probably have done a lot more screaming and cursing than she has. And so the debugging process is it is a process that everyone goes through, but hopefully at the end, when you do end up with a result, it keeps you motivated to keep going on. And so just as the final reminder that, yes, there is an entire community behind all of this, all of the materials that I have, including the artwork that I'm presenting, these were all created by people in the R community. And so just know that we all are all there to help you learn this material. And so just for the expectations for today, and it's really going to be today and Thursday, there is a lot to cover under a very little amount of time. And so the expectation is I don't expect you on Wednesday or on Friday to go into your lab or, you know, start doing your entire analysis in R right away. It is going to take time, but hopefully at the end of this 2-day series, you get a better understanding on how to curate your data. Even if you never program with R afterwards, when you're setting up your next data set, hopefully you pick up some of the tricks and tools and methods. So if you do hand off your data set to someone else, they can actually help you process your data. And as you learn more about R, you can now have a more meaningful conversation with the analyst, or by doing some of the analysis yourself, since you might actually know the domain better than the analyst, you can have a more meaningful conversation. Okay, so with that, we can start our first chapter on spreadsheets. Okay, so spreadsheets. Hopefully everyone has a spreadsheet program, whether that is Microsoft Excel or one of the free and open source versions, LibreOffice, or I think it's called FreeOffice is the other one. So hopefully you have some way to open up a spreadsheet program. And the first exercise I have for people is here are 3 potential patients, Dena Doom, Priscilla Holding, and Anya Warner. And you are taking vital signs during a randomized controlled trial, and so you're monitoring their vitals. So in the spreadsheet, what I want you to do is take those 3 people and input those values into the spreadsheet program in any way, shape, or form. And I would even say it doesn't even have to be one sheet. It can be multiple sheets. Just pretend that you are there to collect data and then enter these data points. We'll give you about 3 minutes to work on that. And when you're done, click the green yes check mark. Oh yes, I forgot to mention, you can always use Google Sheets if you don't have the an actual like program to do this work in. So if you're still actively working on this exercise, can you just put a red X so I can tell if the people who don't have green checks, if they're still actively working or they're just not actually doing it. That's fine if you're not actually doing it. I just need to know if I need to move on to the next portion. Okay. So I'm going to clear all of the Zoom participant responses and go through a few potential solutions for this exercise. So one of the first things that we could have done is take all of the numbers and put them into the corresponding column, right? So without any notion of any units or anything like that. So for example, we have a column called name and we put the person's name in, we have a time, we have their entire blood pressure in one column, their heart rate in another, et cetera, right? And so one thing to think about when you are entering data sets is certain units are going to be understood, like heart rate is probably going to be beats per minute. Same with your respiratory rate, et cetera, but then you have the notion of like what unit is temperature being measured in. And so, yes, there is a quick way you can check by saying that like, well, if the temperature is above, essentially 40, we can assume that that is a temperature being read in Fahrenheit, not Celsius. But that is something that you want to think about. It's going to be pretty, one of these days, you're going to have a data set and you're going to have temperature ranges from like in the 30s up into the hundreds, right? And that is another data processing step that you're going to have to deal with. So especially if you are curating your own data set, that is something that you want to be able to think about is what units is this information encoded in? And so one way we can fix that is we can literally put the unit information straight in our column, right? So we can say pulse ox is a percentage, so we can say 98%. We can say temperature, this is 96.8 F, and we can put the actual unit information in there. But now, one of the things when you are working with data sets that you want to keep in mind is each cell in our data should only contain one bit of information, right? So for example, even if we look at blood pressure, we have the systolic and diastolic pressure all in one column, right? So the whole thing is a blood pressure reading, but there's actually 2 bits of information in there, right? So if our task later on is to like, what is the range of systolic blood pressures, we can't actually do that very easily without going through more of the data processing. Same with temperature. Now that we have put temperature in the unit itself, in that value, that column now contains 2 bits of information. It contains the actual value, and then it also contains the unit, right? So it would be good to, if, for example, we do have a scenario where we might be mixing Fahrenheit and Celsius, you could theoretically filter your data set based off of Fahrenheit and then calculate an average, and then filter your data set on Celsius, do the conversion, and then add that to the average or something like that. Because as it stands now, you can't really just pull the values in a column and do something with it because there's 2 bits of information that you need to be able to work with. And so again, keep in mind that when you are assembling your own data sets, each cell should only contain 1 bit of information. And so just to go on with that, what you can do is then split the name into more pieces, right? So just by creating more columns isn't really an issue if it makes sense, right? So, before we had our name in 1 column, but now we can split it out into 2. And somewhere in this example, I believe I also split out the temperature reading in its own separate column. One thing that you can do is place the unit information in the column names themselves, right? So we can be really explicit and say blood pressure is a BPM, heart rate is a BPM, temperature is an F. And that way, if you are entering data manually, you can sort of understand that like, okay, this column is all Fahrenheit and I don't need to worry about Celsius. And if I am working with someone who works in Celsius, then they would know to do that conversion before they enter that value into the data set, because that's sort of what that column represents. You do want to be careful when you add a lot of information or special characters in the column names. That is something that you want to be able to work with. Like, for example, the percent sign is really like a special character. You sort of want to avoid those. In general, if you do have the ability to create your own data sets, I would highly suggest you keep everything in lowercase. And if you need a space, you use an underscore. And we'll see more about how that plays in when we start reading these things into a programming language, because the underscore actually, the space actually is one way programming languages separate terms. And so it won't understand like heart space rate as one term. It's going to understand heart as a term, and then you gave it a second term called rate. That's sort of why sometimes when you get data sets, why are things like they have dashes and underscores instead of spaces? Because visually, without having the space and having capitalization, looks nice. But as soon as it gets read into a computer, now you have to make sure that you type in a capital H for heart rate and not lowercase h, because that matters. Now you have to know that this is a space, and then you have to write extra characters to tell it it's a space for one word versus a space as a delimiter. There are sort of conventions that people sort of migrate towards, which is keep everything lowercase and use underscores. But that will vary depending on different systems that you use. But that is something to keep in mind when you're putting together your own data sets. And so one possible solution that isn't perfect is, we have diastolic, systolic separated out into separate columns. We have heart rate and respiratory rate. These column names are all lowercase with underscores instead of spaces. We have a temperature column, and then we have a separate column for your Fahrenheit or your actual unit, right? And this isn't going to be perfect. There's no such thing as like a perfect data set, especially if you're curating it on your own. But it is something that I think about because I know what the next series of steps could be. And so I'm going to prevent myself from work later on by sort of handling the data input as nicely as possible. So there's less cleaning involved later on. And so the main takeaway here is really only put one thing in each column, right? So every cell in your data set should really only contain one unit of information. And the more you sort of separate that out, the more flexible your analysis pipeline later on can be. Okay, so another exercise in the same, we'll build off of the example that we just had. Don't worry if you don't have to sort of swap your data sets around to any of the quote unquote better examples. But let's say you have two more readings. So you have a reading 12 hours on the same day, and then another reading taken 24 hours on the following day. And so let's take another 3 minutes to go through entering that set of data into your spreadsheet program. And again, put the green, put the green check mark when you are done. I don't know where to put this thing. I'll just keep it off the side. And you put an X if you are, if you need help. And if you have any other questions about what we just talked about, you can totally mention them in the chat as well. So let's talk through some of the examples for this question. One possible solution that you could have is you create a separate table for each patient, right? So you can either have in as a separate sheet all of the values for Gemma Dune, or in a separate Excel file for each of the 3 patients that we have. And so actually, I realize I understand why that screenshot looks all the same is because that table doesn't actually read off of a data set. Anyway, so there's one way that this would make sense. Let's say that your study is completely individual patient focused, in which case it would make sense to have one separate file or one separate sheet for each person that you're working with, right? It makes it really easy to essentially make sure that the data for each patient is collected properly. It's a little bit easier to go back and check if something was done. If you are just looking for specific values for a specific person or to compute some sort of statistic for one person, it becomes an easier task. So that is one way that you can set up this data set is you have one table for each person. The only downside is if you do go this route, if you need to start saying, for example, what is the average heart rate for all of my patients, then you have to realize that then there is going to be a data step where you combine all of your data sets so you can do that step. So the main takeaway here is as long as you have some rationale for how you're going to set up your data set and then understand that later on you need this one other step, then you're okay. Another thing that's more common that I see in Excel when working with spreadsheets is instead of creating a separate file or a separate sheet for each person, you start using white space as a visual separation between groups. Right. And so you can see, let's say, for example, one way you could have done this is you have one person in one set of data, and then you have another visual break somewhere to designate another person. And that has the same benefits as before in the sense that yes, you're easier to see if I'm expecting 5 values for each person, you can count 5, 5, 5 or 3, 3, 3. You can still do your little one-off calculations if you need to. But then the only issue is when you start working with this data set, you have to deal with these blank rows if you start loading them into a program. And also this type of formatting sort of makes it almost too easy to make these one-off calculations off to the side. You can see, for example, let's say I make a calculation of the average diastolic heart pressure for Anya, and I make that calculation underneath her values. It's almost too easy to say oh, now I want to calculate the average for everyone, and you pull that whole column. Now you have that value in that column already. And so you want to make sure that you aren't really doing these one-off calculations to the side, because that is then part of that rectangle that is your data set. So another way you could have done this, and this is sort of instead of stacking your patients one below the other, you can stack them sort of next to each other, essentially a column-oriented formatting. And that sort of solves the problem in the sense that I still have my people by groups. The first 5 columns is Dena, the next 5 columns is Anya, et cetera, et cetera. And I'm not using white space as a delimiter anymore, but then I still have the benefit of checking values and stuff like that. The only problem with this notation is you're going to have definitely repeated column names if you follow that kind of formatting, which can be a problem later on. So let's say you want the first name column, you'll get 3 of them back, or some programs don't even allow you to have duplicate column names. So that might be a problem later on when you're trying to work with your data set. And if you paid attention to the news within the last month, the people in the UK got into a lot of trouble doing this type of data formatting, where they started putting data into a column-oriented format, and they literally ran out of columns in Excel and lost a bunch of patient information because of that, right? So be mindful when you start thinking about adding a lot of columns. So that sort of is related to that first exercise, where it's like make as many columns as you need. That's still true, but you don't want like groups of data stored in columns in that format. So there is sort of a distinction there when you're putting together your own data set. And so one way that sort of I would suggest you work with your, like when we're creating that raw data set that we can do our analysis from, remember that little Likert scale that that I showed you from our results. One way you would do this is sort of take that format that I presented in exercise 1, and then just continue on entering values in that order, right? So for me, I sort of pick like chronological order for like day. And so what this will allow me to do, like even in Excel, I can use Excel or any spreadsheet program to do some kind of formatting or filtering that way. If I need to, sort of sort by first name and last name, I can still kind of do like visually see if I have the correct number of people for each group, et cetera, et cetera. But this is really the format that really, like if you try to load it into any piece of software, like this is the format that it needs. There aren't like extraneous white spaces around. I don't have repeated column names because I'm not stacking my data column wise. They're all stacked row wise. And then I have pretty much like one unit of information per each cell. Right. And so the main takeaway from the first 2 exercises is, as long as you have some rationale for the way you're setting up your data in the beginning, and you have a plan for how to deal with it later on, there really isn't a messy data set, right? Like we get messy data sets because it's usually like someone else's data that they give us and they didn't account for different things. And so now we have to go through the process of like extracting out values, getting rid of units, stuff like that, or converting units, et cetera, et cetera. And the other takeaway is, yes, always pick the data format, especially if you're manually entering data. Pick the format that makes your manual data entry easier. And then on Thursday, I'll show you, like, as long as you have a plan from your manual easy data input, there's very quick ways to get it into a format that you can actually start doing the analysis with. Right. And so the last exercise really is, okay, let's say I am asking you to identify bad, quote unquote, bad values, right? So blood pressure greater than 140/90, resting heart rate greater than or equal to 90, your respiratory rate's over 20, your pulse ox is under 95, your temperature is over 100 degrees, any one of those values. How would you just take the data set that you have now that's in front of you, and then how would you mark that information? And so we'll take 1 minute, since there isn't really too many like bad values. So just put a green check mark when you're either done with the exercise or you're just waiting around. Click the green check mark when you're ready for me to move on. We'll put it that way. But I'll give everyone about a minute. And so this exercise is really focused around, we have a data set and we know that there are like values that we need to look into. How would you actually flag those values or those observations for within our data set? So one way you can do this is literally color code the values that are bad. So in this example, any of the values that are bad, I can flag with a red color, for example. Or any of the values that are good, I can flag with a green. So spreadsheet programs usually allow that. So the problem now for that particular observation or value that is bad, we sort of violate like that first rule of each cell containing one bit of information. Because let's say, for example, in my use case, like this 142 diastolic pressure, that is a bad value and I'm coding it red. And so now that 142 holds not only the value for the blood pressure, but also the fact that it is a bad value. Values two things wrong, two things encoded in that particular cell. One thing that I've actually encountered in the past was I've encountered a data set where the color red was how the data set denoted a negative value, not with a minus. Loaded that data set in, I don't really see red as like all I see is the actual number. And this was a negative number other than the fact that it's red in the label. And so sort of the extreme case of what can go wrong when you put too many bits of information in one cell, it makes the data set less portable, but it also, you have to now do additional work to take apart all of those bits that's being encoded. Have only two rows that have values that need to be reported. And then what I can do is use either the spreadsheet system or in R, and I will show you how to do that in the next hour, filter all the rows such that the report cases needs to be yes. And then you can get a nice, pretty report. And this makes it better because now I can filter my data set very quickly and then write out another data set that's essentially like, these are all the values or people that need to be, you have to send an adverse event form for. And you're not relying on just colors because you might, like, colors can be taken out or removed. It's really easy to select all and remove all the formatting, in which case you've lost that bit of information. This way, the actual flag of whether or not that case needs to be reported is actually part of the data set itself and not like some visual artifact that you're looking at. And so the main takeaway is treat your data set as an actual piece of data, not as an actual data visualization, right? So adding things like color, adding spaces, that all are you working on a spreadsheet program and trying to use the spreadsheet program to give you as much information in one go as possible. But in that case, that's what visualizations are for, not actual data sets. So data sets are used to create the visualization, but don't treat your data set as the visualization itself. And it's hard if like Excel and spreadsheet programs are the only thing that you've ever worked with, because that sort of enforces and encourages you to use your data set as a visualization tool because it's always in front of you all the time. But as you sort of move on, you'll realize that if you treat your data raw as its original unaltered form and you create the visualizations from it, your data set and your analysis is going to be more portable, and you don't run the risk of having too much information encoded in a data set that needs to get taken apart later on. And you also don't mess with the actual raw data set. And so you can have your data set update and then run your entire analysis pipeline and end up with your visualization. You can imagine, like, let's say you go through this whole process of like flagging individual cells, and then you get another data set that replaces the old one. Now you have to go back and flag it all yourself again. And so that's also a lot of work because you're modifying the raw data set directly. And so the last thing, this isn't really like an exercise that I'll have a break for, but let's say you examine the patient and you ask them like, why is their blood pressure so high, and they're like in the room like doing yoga, right? And so there's always going to be like some kind of column that's really just like bunch of notes that essentially just say like a patient was just doing yoga. That's why this is not really an adverse event, even though it met like SAE or AE like results to report. Right. So that's all we have for the first unit on spreadsheets. So just as a summary, spreadsheet programs serve as a graphical user interface to one's data. It makes that really simple that like we can see the data set and point and click and interact with it. These GUIs are very convenient, but they come at a cost of flexibility, because when we try to load that stuff into a programming language to do more analysis, then we have to deal with all of the one-off calculations, the spaces in our tables, stuff like that. But when you're putting together a data set on our own, it is one of the most convenient ways that we can input data. So if we're trying to collect data, like we want that process to be as frictionless as possible. And so spreadsheets do have a place for that, but just be mindful of the next steps when you're working with your data set. Okay, so let's take a 5-minute break, put up a 5-minute timer. And if you have any questions, just let us know, but we will just reconvene in about 5 minutes. Okay, so we are now going to start the process of working with R and RStudio. And so hopefully no one had any installation issues. I didn't get any emails or no one really showed up to the office hours, so I'm okay. I'm assuming that everyone has RStudio and R and the Tidyverse package installed on their computer. There's a few things that we sort of want to go over before we start programming. This is the RStudio panel. When you open it up, there are, by default, when you open it up, there are 3 panels shown. The left-hand side, where there's the console, terminal, markers, and jobs tab. This is where actual R code is going to be sent and run. The top right corner under the environments tab is probably when you are starting off actually not even when you're starting off, like this is probably the tab that you're going to have open the most. What's nice about the environments tab is any data set that you load in, any type of variable that you're working with, they will all be listed here. So it's a really good way to make sure that like if you created a new column or if you subsetted your columns into another data set, that it shows up here. On the bottom, under by default, it opens to the Files tab. And this is the regular files browser on your computer. So what we can do is when we're executing our code, we can, if we wanted to, type something like 3 + 3 into the consoles tab, and we're essentially using R as a glorified computer. But this isn't useful in the sense that like all of the commands that we type aren't saved anywhere. And so the way we can create a file that we can save is we can, in the top left corner, you either can go to File, New File, and RScript, or there is this white piece of paper with a check with a plus on it. You can click that and then go to the first option, which is RScript. And what you'll now have is a 4-panel system where the console panel gets split into 2, and then on the top, you can type in your R commands. So let's say 10 + 10. And then the way we can send the code that we write up here down into our console down here, one way you can do it is simply copy paste the values that you want executed and it'll go down there. The other way you can do it is select the rows that you want executed and you can hit the keyboard shortcut, which is Control-Enter. And what that will do is that current line that your cursor is on will be copied and pasted into the console down here and then executed. There is a little button up here that says Run. And if you hover over it, you'll see that it says Run the current line or selection, which is also Control-Enter. So if you forget the Control-Enter keyboard shortcut, you can select the bits of code that you want run and then literally click the Run button. And what it will do is it'll send the code from up here down there. A few other things that you want to set up when you are working with RStudio is under the Tools panel on the top. If you go to Global Options, here, what I like to do is the first 2 checkboxes up here that says Restore Most Recently Open Project at Startup and Restore Previously Open Source Documents at Startup. These are optional to have them on or off. I personally like it all off. That way, every time I start up RStudio, it's not opening anything that I worked on before. And so that sort of is one way to prove to myself that I'm starting off in a clean working area or a clean working slate. The part that you actually use to guarantee that every time you start up RStudio, you don't have some random data set lingering in the background is this first checkbox that says restore.r data into workspace as startup. You want to make sure that is checked off. And then the drop down underneath that says save workspace to R data on exit, you want to set that to never. And what this will do is when you close RStudio, all of your data sets that you have loaded will not be saved. So it's forcing your code to be reproducible in that sense. And then the next time you start off RStudio, it's not going to automatically load all of those things into your environment. And the benefit of, and the reason for that is, one, let's say, for example, you loaded in a data set that's very large. The next time you open up RStudio, it's going to try to reload that data set. So you're going to end up waiting in front of your computer for like 5 to 10 minutes just for RStudio to work. So you don't want that to happen. And another thing is, let's say you're working with like 2 different projects. Both of them, you created a variable called patient. You have a patient data set. If it automatically loads up, you might be confused as to which actual patient data set that you're working with. And so you want to make sure that all of that stuff is clean off the slate. And that's more of a recommendation that will help you. It will solve a lot of problems, especially when you're starting off, when you are learning, when there's all this information of like, what do I have loaded and what do I not have loaded? If you just start off with a clean slate, you can at least be sure that that's something you don't have to worry about. And then click OK at the bottom to save those changes. Okay, so the next thing that we're going to do is we are going to download the projects or the data sets that we are going to use for our lesson. And so this is one of the lessons. I'll put this into chat, and it's also at the top of the Etherpad as well. If we go here, and on this page, towards the top, there is a button called Code. And if you click on Download Zip to download the zip file, you can download this code repository down to your computer. The most important thing is this data file and this R project file, right? And so what I'm going to do is I'm going to click Download Zip. I'm going to save this onto my computer. I'm going to put this on my desktop. And then we will extract, unzip this folder on our computer. And what we should see is the data folder and this R project folder on our desktop somewhere. So can I get a green check mark if everyone has this file on their computer somewhere? I'm putting it on my desktop because that's a little bit easier to find. And if everyone does it, then it's easier to figure out if something goes wrong. And put a red X if you are having issues. Okay, so now that we have this file on our desktop, we can go back into RStudio. We'll talk a little bit more about this in the next chapter, but the way we can like quote unquote open this project is on the top right corner where it says project none. We can click this and then go to the second drop down that says open project. This is because I have an empty file, that's fine. And then we're going to navigate to that folder that we have, and we are going to pick that file that says ds4biomed.rproj, so that R project file. And what that will do is it will open up this entire folder structure. It essentially gives us a new R session. And the most important thing is on the bottom right corner, you should see at the top where it lists the folder that you're working in. It should be that folder that we just unzipped and did our work in. On the top right corner, what you should see is the name of the project. So for me, it says ds4biomed-learner. And so that is the project that we're in. Okay, so once we have that, we're pretty much ready to start loading up our first data set. And so there's a few things that we need to sort of understand how things work when we want to learn our first load our first data set. The first thing is, if you're brand new to R, we are going to be using a collection of R packages called the tidyverse. This is sort of a teaching decision that was made to make things a little bit easier. So if you know how to work with R in the past, this is sort of what we are using. The tidyverse is a collection of packages that have a really consistent way of working with your data set, which makes it a little bit nicer as a first-time teaching way into R. And it comes with packages to read Excel files, read CSV files, pivoting your data sets, and it's part of a larger ecosystem that also feeds into fitting models and stuff like that. So the first thing that we're going to have to understand is finding our files, right? And so let's say, for example, we are in this ds4biomed folder, right? So on the left-hand side, it's going to be on Windows. On the right-hand side, it's what it looks like on a Mac. And mainly the only difference is Windows, the beginning of your file starts with C:\\. So it's like your C drive or whatever drive that your desktop is in. And then on the Mac side, it's a regular slash, meaning like this is the beginning of your computer. Correct. So you need a separate package that's included in the tidyverse. So that's part of the reason why we're going with the tidyverse, because it's one install and it installs a whole bunch of things for you at once. So you can actually use a library within the tidyverse to read in an Excel file. And it can be an Excel file with multiple sheets. So you can actually read in a specific sheet from an Excel file into R. And we'll go through that example right after I show you how to load in a data set. So yeah, so all of that stuff I talked about with the spreadsheets, like you can do all of that work in Excel and then you can load all of that information into R, and that's totally okay. It's a little bit harder to write out an Excel file. It needs a little bit more installation stuff, but you theoretically can as well. All right, so back to finding our files. Let's say we are in this ds4biomed folder. This folder that we are in is known as the working directory, right? So what is the working directory? In our case, it's going to be C drive, users, Daniel, desktop, ds4biomed on Windows, and then it's going to be / users, Daniel, desktop, ds4biomed on a Mac. If our task is, hey, from this folder that we're in, how do we specify this medical data tumor growth data set? The way we can specify that, there's one way we can specify it is the relative path. So because we know where we are already starting from, we can say from the ds4biomed folder, go to data, and then go to medical data _ tumor growth.csv, right? So we can specify data / medical data tumor growth.csv, right? And so that's the relative path, because we are specifying that file location from where we are starting, which is our working directory. And then there's another way we can specify this file, which is give me the absolute address, right? So one way you can reference like relative path and absolute path is like, if I want to go to the Starbucks that is literally around the corner from me, I would leave my apartment, make a left, and I'm there. And so that's the relative path. Or I can give it the absolute path, which is the actual like mailing address of that of the Starbucks, right? So we have the absolute path for this file as well, which starts from the very beginning. So on a Mac, if you ever see a file path that begins with a slash, then you know that that's a path, that's an absolute path that is starting from the beginning of your computer. So it's going to be users/user/Daniel/desktop/ds4biomed/data/medical data_tumor growth.csv, right? And then the same thing for Windows, except it's going to be a C:/, right? So yes, the absolute path takes more typing, but sometimes you just want to be like, I want to load up that data set and it's like some ungodly place on my computer. Sometimes it's easier to specify an absolute path, but for the most case, we try to work in relative paths. And that sort of helps makes your data analysis more portable if you work in relative paths. And so the thing that makes relative paths work is this understanding of a working directory. You have to know where your computer think you are, right? Like think of it as like when you're double clicking through your folders, your working directory is the current folder of stuff that you're looking at, right? And so the programming language that you're working in has to understand like what is the folder that you're looking at, so then I know where the program is, and that's really important. So let's say we move our load data file into a folder. We create a folder called analysis, and then we put in our file there. How do we specify this medical tumor growth.csv data when it's not like double clicking into a folder, but we actually need to go one folder up, right? So if we look at this diagram here, there's this ds4biomed folder. It has a data folder, an output folder, and analysis folder. In the analysis folder, we have this loaddata.r file. So this is where we are now. How do we specify this file from there, right? So our working directory now is this analysis folder. And the way we can specify this relative path, how do we go one folder up, like clicking the little up arrow or hitting the backspace when you're navigating your file system, is with these two dots. So from the analysis folder, if we say .. it means go up one folder. So now we're in the ds4biomed folder, and then back down to data, and then back down to medical data, tumor growth.csv. So that's the relative path. But at the same time, our absolute path never changed, right? It's still this entire thing from the very beginning. So relative paths are always relative to where you're starting from. If you need to go one folder up, it's using two dots, and that's how you specify the previous folder. And so here is a quick example. You can type the solution into chat when you're done, but here's an example of a folder structure. And let's say you are here in this analysis folder, you're working on this demographics.r file. What is the relative path to the RCT patients.csv folder down here? So that's question one. And then question two is, what is the absolute path to the chart review data patient CSV? So there's two CSV files called patient. And our current working directory is this analysis folder. So, and then I want you to specify relatively, how do we get to this RCT folder? And then absolutely, how do we get to this chart review folder? So we'll take about 3 minutes. Put a green check mark when you are ready for me to move on. But if you have a solution, paste it into the chat, and then we can discuss it. And just so you know, I can also, if you have a question, you can also type the question out to chat when we're working on exercises. So I can try to address them as well, if you're comfortable with your question out in chat. Okay, so let's go over this question. So question number one, what is the relative path to the patient CSV file from in the RCT folder, right? So let's say we're in this analysis folder. How do we get to this RCT folder? So the first thing we need to do is go up one folder into chart review. And so the way we do that is say .. And then we go up one more folder into the projects folder. And then we can go into the RCT M220305 folder, then specify data, then patients.csv. Right, so the answer for the first question is how do we specify a relative path to this other folder? The trick is using .. to go up one folder, but we can go up multiple folders in a row, just like double clicking. You can think of these slashes and these .. as us double clicking on the folder. And the next question was, what is the absolute path to the patient CSV file in the chart review? So the absolute path is going to start from the very beginning, which is /, and then it's going to be users, Daniel, projects, chart review, data, patients.csv, right? So these are two ways that we can specify a file on our computer, and this is the basis of specifying the file that we will use to load into R. So I will paste those solutions into chat, just so you see it. There's one more caveat for paths in Windows. In general, there is that / in Windows, but on a Windows machine, backslashes actually stand for a really special character from pretty much every programming language out there. And so on Windows, even though when you look at it on your computer, it's using that /, feel free to use the /, which is the key that's attached to the question mark, not the key that's above the enter key. So that's probably a key that you very rarely hit unless you're programming, but it's the key with the question mark. You can use that as well, even though on Windows computers, when you're clicking through the system, it's showing the /. You are free to use the /. If you do use the / because you're copying and pasting Windows paths into a programming language, you have to put 2 backslashes. So that's sort of the annoying thing with Windows, but this is just how it is. And then the question that we were talking about is like, okay, what is our working directory? I was talking about what is that folder that we are constantly working in? That's what setting up the R project folder did. When we went to that top right corner and opened a new project and navigated to that folder and then selected this ds4biomed.r project file, that is Rstudio's way of setting your working directory. Your working directory is not just what's displayed in this corner. And that's why I highly suggest you to use R projects, because then it is the projects folder, which is what you will see on the top right corner of Rstudio. Okay. So now we can actually load up our first data set. What does this look like in R? So we can open up Rstudio. And I can click the top left corner and go to new file. So we have this new file. And just so we have access to all of our notes and code moving forward, I can save this file. And I'm going to save it in the same location as where this ds4biomed file is. And I am going to call this 01-load data and hit enter. And so another thing that's really useful is when you are naming your files, adding a number at the beginning so that your files sort in the order you want to load them in or look at them is a pretty good trick. So if I have a file called load data and then analyze data, the A is going to be sorted before the L for load. And so the way I fix it is I prepend a number at the beginning. So it's like 0, 1 load, 0, 2 analysis. And that way, if I go back to this project like a month from now or even tomorrow, I know the order I need to look at my code or do my analysis. And this is one way you can slowly start setting up pipelines. Okay, so now let's actually do something in RStudio. We are going to use the tidyverse set of packages. And so the way we load up those packages is using this function called library and then tidyverse. So I will paste this in chat. So if we say library tidyverse and then hit control enter to run it in our console, you'll see that at the bottom, it's going to start attaching packages. So tidyverse, again, remember I said it's a collection of a bunch of other packages. So these are all of the other packages that got loaded. And then at the bottom, it just says down here, certain functions share the same name, and I'm just letting you know these functions share the same name. But we won't be using any of these functions in the next 2 days, so you don't have to worry about that. What we should see, and this is what you should see the very first time you run it. If you were to run it again, what you should see is an empty reply. What you should not see is something along the lines of, like, if I type this wrong, you would see something like, there's no package called blah. And then it's either you type the library wrong or you don't actually have the library installed. And so if you do have a problem with there, do let me know, because we do have a backup system that you can use to follow along. Okay. So now that we have the tidyverse open, there's actually a function. So if you got the warning that was tidyverse was built under our version 4.03, that's fine. That typically you get those warnings when you updated R and then you installed tidyverse like previously in the past, and essentially saying that those two versions are not aligned. You don't have to do it now, like don't do it now, but if you were to reinstall tidyverse, that should fix a lot of other problems. That should fix that warning. So now let's load up our data set. There is a function within the tidyverse called read_CSV. And so what might you think this would do? It will read a CSV file. The way we specify this is read_CSV and a set of round brackets. So we're calling a function called read_CSV. In here, we put in quotes. You can use single quotes or double quotes. It's either one is okay, as long as you begin and end with the same. And then we specify the file that we want, right? And so that's what that entire exercise before was about. How do we specify files? How do we find files? We can find a file relatively to where we are now. Because we are in the ds4biomed folder, I know that there's a file in the data folder called medical data tumor growth.csv. So we can specify that file by saying data/. And the nice thing when you're specifying files is if you do this and you hit the tab, like the tab key, like literally to the left of the letter Q, it'll try to help you along and say like, okay, you specified the data folder. Here's all of the files in the data folder. Now you can actually use your arrow keys to navigate to the file that you want. So medical data _ tumor growth.csv. And this will it's one way. If you don't see the thing that you're looking for pop up, you're probably in the wrong location. That's one prompt. Or two, you spelled something wrong in the beginning or somewhere along the lines before you got there. So using the tab key is a really good way to essentially type faster, but also not make spelling mistakes. And so when we're done with that, we can hit Control Enter, and that CSV file would have shown up down here at the bottom. Right. And it's going to say a bunch of information like parse columns with specifications. And it's saying like you have a group column, a GRP column, a group column ID, day, size. And then this is a character, which is like a bunch of letters, a double, which is a number, a double, which is a number. So it's a column that contains letters and numbers, and then a bunch of columns with numbers in them. And then it dumps us out the actual data set at the bottom. There's this group column, this GROUP column, ID, day, and size. And so that's what we do to load up a data set, right? And if you end up with something like could not find a function or does not exist in current working directory, try to make sure that you spell things correctly. That's sort of the first level of things that can go wrong. Okay, so that's how we can load up a data set, but it's not useful if every time we want to work with the data set, like we want to build on the, oh yes, and like it is all case sensitive. So hitting the tab key will also make sure you don't accidentally put in a capital letter when you didn't mean to. This is sort of the place where having underscores and spaces slowly become a thing in your file names. So this is the code that we use to load up a data set, but it's not helpful if every time we want to build on this data set, we have to reload it. And so we can save this data set that we loaded into a variable. So I'm going to copy and paste that code again, and we can save it into a file called, let's call it tumor. We can type tumor space, the less than symbol, the dash, and then space, and then read underscore CSV, that read underscore CSV command, right? So I'll paste that into chat. This less than dash is our way of assigning the thing to the right, the value, R will execute the thing to the right, which is our read command, and then assign it to the variable to the thing on the left, which is tumor. And so now if we run this command, the first thing you'll notice is the output only tells us the column names it read out. It no longer dumped out the actual data set. And that's because if you look at the top right corner, that data set has now been loaded into a variable called tumor. And here, this is when the environment panel becomes useful because it tells us it's got 574 observations with 5 variables. And so now if we only type in the word tumor and we run that bit of that code, that's how we can see our data set. Right, so now anytime I run the word tumor, it's going to be referring to that data set that we loaded, so we don't have to constantly reload our data set. Just to round off this example, there is a function called read underscore Excel. Part of the tidyverse, it comes with a library called readxl. And this is the library that you need to read Excel files. So we can take the same piece of syntax as before. So let's call this tumor underscore XL, less than dash to assign a value. So in this case, like the spaces don't really matter, but they matter in the sense that just realize I have my white space showing, and that is so the spaces don't matter, but it's really useful because you don't want this dash here to be accidentally mistaken as like a negative sign when you're trying to assign numbers into things. And so it's a good habit to put spaces around the assignment operator. That's what this is called. And then we have a function called read underscore Excel. And then in here, we can pass in a file to the Excel file. And so this data set actually comes with an Excel file. It's under data / and it's we can call this medical data tumor growth .xlsx. And you can see that is an Excel file that we loaded into R. And both things show up in R in the same way. And this object that showed up, whether we use read underscore CSV or read underscore Excel, is called a data frame. Right? So once you have a data frame object into R, all of the data manipulation steps that we'll be covering today and on Thursday, they all apply. So most of the work is actually how do we get this data set into R as an R data frame object, and then once it's there, then we are then you're good, right? So there's a lot of work of getting all like SPSS files, SAS files, Stata files, like they can all be read into R. And then from the R's point of view, it gets converted into a data frame object, and then you can continue on. And so how do we know what is this object or this thing that we're looking at? There's this function called class. And if we give in anything that's a variable in R into class, so if we say class, open brackets, and then tumor, you'll see at the bottom, it's going to say something along the lines of like Tibble df, Tibble, and then data.frame. And that's how you know you are working with an R data frame object. Okay, so we'll take 1 minute to work on this exercise. Make sure you have the ds4biomed Rstudio project open, load up, and the actual exercise is load up the exercise-01-050.csv under the data spreadsheets folder. You can either use a relative path or a full path, and then save this to a data frame object called spreadsheet underscore df. And then if you need, there is a sort of a code snippet down here that you can use as a reference to this assignment. So we will take 1 minute, put a green check mark when you have this data set loaded up. Yes, I saved the Excel file as tumor underscore Excel to distinguish it from reading it as a CSV file from reading it as an Excel file. So that's the reason why I called it tumor underscore Excel. Part of it was to show you, or you can see for yourself, that even though one came from a CSV file and one came from an Excel file, from R's point of view, they are the same. Okay, so let's go back to R. And so one thing I forgot to mention, in the top right corner of this environments panel, if you click the little thing that looks like a spreadsheet over here, it will actually open up the full data set for you to view. So you do get like sorting, so you can click on the columns to sort. You can click on filter to filter by view. So you do get some sort of like spreadsheet functionality to look at your data set. And this is useful for if you're like, did I load my data set incorrectly, or you're just looking for specific values without needing to without needing to write R code. This is a pretty quick way to do that. Just make sure that you close it, because as you make changes, that view, that tab that shows up on the top doesn't actually refresh itself. At least I don't think it does. Maybe they fixed it. So the exercise was to read up that exercise file as a spreadsheet underscore df. So the way we do that is we say spreadsheet underscore df. The way we pronounce this operator is gets. So spreadsheet underscore df gets read underscore CSV, and then we specify the data set, which is under data slash spreadsheets slash exercise 01. I just picked any random one. And so now I can click on this, and this is literally one of the data sets from the examples that we had. Okay. So we have our tumor data set, TUMOR. So this is the data set that we loaded from our CSV before. So you have a data set. There's a couple things that you want to do when you're first looking at it. First, print it out. by literally just typing it out to make sure that the columns got read correctly. There is a function in read CSV to say skip how many, like let's say your data set starts on like the 10th column, the 10th row. There's a way to say like skip the first 10 rows and then start reading your data set there. And this is a good way to say, to make sure that your column names are read in properly and you don't have column names that's like actually the first row of your data set. So one of the first things that we want to do in our data set is we want to be able to select or filter or subset our data set by the columns. So the way we do that is there is a function called select in the tidyverse. So after you make sure you library the tidyverse first, there's a function called select. The first thing that you pass into select is the actual data set you want to select from. So in our case, we want to select from the tumor data set. And then we put a comma, and then we comma delineate the column names that we want to select. So let's say we only want the size column. We can say comma size, and because size is a capital S, we have to make sure we specify it with a capital S. And now if we hit Control Enter to run that, we get our data set that is just the size column. And so we can do that for if we want to reorder our columns or select multiple columns, it's all through the select function. So we can say select tumor, comma, let's say we want size first. So we can say size and then group, comma, day, all with capital letters. And then you'll see that we are selecting multiple columns and in whatever order that we want. And so if you ever need to filter columns based off of, select your data based off of the columns, then you use the select function. And this is really useful for, especially when you're working with, what is it called? Survey data. And you only care about like the person ID and like these 5 questions. You can load in your data set and then select based off of only those 5 questions. So you're not having to like scroll through like the hundreds of questions involved. So there's another way, if you've seen tidyverse code before, there's another way you can write this code. You see that the first function, the first value in the select function is the data set. That's pretty much going to be the case for all of the tidyverse functions. Like the first thing that each function takes is the data set. And the output of the select function is in itself a data set, right? So we can rewrite this by saying tumor, T-U-M-O-R. And then there's this pipe operator, which is percent greater than percent. So you hold the shift key and then you hit 5, period 5, essentially. So it looks like that. And then we can rewrite the top function that's selecting tumor size by saying tumor pipe, or the way we pronounce it is the word then. So tumor, then select, and then size. And this does exactly the same thing as the previous example where we just selected size with tumor and size. Then we can essentially rewrite it by saying pass in the value of tumor into the first argument of select. Select takes a data set and a column that we want to select. And so the column we want to select is the column called size. So if we wanted to rewrite the second example, we can say tumor, then select, I wrote size, group, day, right? And so these two things are, from R's point of view, equivalent. One thing that you see people do is after the pipe operator, they put a new line. And so what this allows you to do is essentially you can read down vertically and you'll get a general sense of what you're doing. So we're working with our tumor data and then we're selecting from it. And so it gives you a linear way to read your code versus inside out way of reading your code. So if you're a mathematician, you're probably used to this functional way of functions within functions within functions. But to read your code, it's actually easier to find the thing on the innermost side first and then read it outwards. And so this piping operator, the way we pronounce that is with the word then, is what gives, that's what it allows us to do. Okay, so next thing, we know how to select rows. Next thing I'm going to show you is how do we filter our, we know how to select columns. The next thing I'm going to show you is how do we filter our rows. With that, there's a function called filter. So filter takes a data set as its first parameter, so we can work on our same data set, tumor. And then we give it some condition to filter on after the comma, right? So let's say we want all of the values in our data set such that the group value has the group column has, the group variable has a value of 1. Then we can say group, so make sure we're specifying the column name correctly. So it's group with a capital G. And the way computers test for equality is using 2 equals. So it's group equal 1 is saying, does the group have a value of 1? If we have a single equal sign, it's saying like set the value of 1 equal to the value equal to the variable called group. So we don't want that. So it's double equals for equality checking. If we run that with Control Enter, you'll see that we'll get, as a response, 97 rows back instead of 564. And then all of these values are just going to have a group value of 1. And so just to prove it a little more, we can say group value of 3, and then you'll see that we have 161 values, and all of the values back, our group is equal to 3. So one of the questions was, if we already loaded a tumor file, you can just use tumor because that's what we set it as a variable. There's no longer a reason to reload that data set because it's saved in as the variable called tumor. Until you sort of use that operator, the assignment operator, to reassign something else to tumor, it will be that tumor data set that we loaded at the very beginning. And one other thing to keep in mind, this file browser in the bottom right-hand corner, this is different from that working directory. This is really RStudio's way of like helping you navigate your file structure without having to like open up like this as a way to open up your file structure. You can navigate your files in here. Whatever you specify in here doesn't automatically switch the working directory for you. So that's sort of why we stick with projects on the top right corner. That's why we use RStudio projects. So we don't have to worry about which working directory we're in. We only need to be concerned with where is this our project file? That's where the computer thinks is our starting point. Okay, so just like I talked about piping, we can pipe this to filter group equal 3, and these two statements that I just wrote are going to be equivalent to one another. It's sort of your preference which one is easier to read. You'll see that when we start chaining commands, this piping notation is definitely going to be more helpful. So we're going to do, let's say we want to filter on multiple things. So we can say filter, and then let's say we want to filter where our group is equal to 3 and our day is equal to 0. And what this will do is an like A and D and operation, such that I'll give you all of the rows where the group has a value of 3 and also the day has a value of 0. And you'll now see that if I do that, there's only 10 rows left in our data set. And this ties back into that first unit of working with spreadsheets, because you can see like as long as you have each column storing 1 bit of information, if you want to do some kind of filtering operation, this is how you can filter it. Because each column only contains 1 thing, you don't have to do additional processing. This is how you can filter your data set, right? So remember before when I was saying like, hey, if the blood pressure was greater than like 100 and like 40, you have high blood pressure, the systolic blood pressure was 140, you can now filter off of that and then save that out, or just look at the values where that condition was true. So this is one way we can say like an AND operation. So I'm going to copy this statement instead of the comma. So by default, the comma is an AND operator, but we can also use the ampersand, which is shift and the number 7, without the comma. And so what this will allow us to do is like, this is the Boolean, like true false way of looking at, like, I want the values where group is equal to, or the group is equal to 3, and also the day is equal to 0. So that's another way of specifying the same thing. And the reason why I am showing you this AND operator is because sometimes, if I copy and paste it again, sometimes you want a condition that is an OR operator. So let's say I want something that's like day is equal to 0, or the day is equal to 13. You can now see I filtered my data set and all of the values where I have a value of day is 0 or 13, right? There's no such thing as a day having a value of 0 and 13. If I replace this with an ampersand, I should get nothing back, right? A table with 0 columns, 0 rows and 5 columns. But if I use a pipe, which is shift and then the key above your enter key, so it's like the vertical bar, probably a key you almost never have used until today. That is the OR operator. And so we can actually now, using those two things I just showed you, select columns and rows at the same time, right? There isn't one function that does both at the same time. We are literally selecting the columns first and then filtering on the rows, or filtering on the rows and then selecting on the columns, right? So let's say we want to take our tumor data set, TUMR, and then we want to select the group, day, and size columns, right? So I'm going from 5 columns to 3 columns. And then we can do this other pipe and then filter it into something like, I want it such that the tumor size is greater than 2000. Right? So now I have 14 values and 3 columns. I selected on the columns that I wanted first, and then filtered on those values second. And this is sort of when the piping operator becomes more useful, because now you can read, if you just look at the word tumor and you look straight down without looking at the code to the right, you can see tumor, you're selecting some columns, and then you're filtering some rows, right? So you get a quicker general sense of what operations there are, operations are happening. And then if you're like, oh, I'm selecting on some columns, what columns am I selecting? Then you can look to the right and then see the code of what you're doing. Maybe you don't care. It's like, oh, I'm doing some kind of filter. What am I actually filtering on? Then you look to the right and say, oh, it's the rows where the size value is greater than 2000. Right. You don't have to type this, but the way you would quote unquote unpipe this is, well, the first thing we do is select. So we would say select tumor group day size. And then we're taking this data set and then running filter on size greater than 2000, right? So R takes this pipe code and turns it into the ones you see on the bottom, but you don't have to write the one on the bottom, because now you're trying to read inside out versus just reading from a straight line. This says tumor, object tumor not found, and that's because I spelled it wrong, TUMOR. And then I get the same values back, and it's sort of up to you what you find is easier to read. A lot of people will say the one on the top is easier to read than the version on the bottom, and I will paste that into the chat window for people. Okay, last thing. And so this is the beginning part of creating pipelines. Is a pipeline, the way pipelines work is there's input, something happens, and then there's an output, right? So I got a question as, So yes, the question is, if you have selected something, you can only run the analysis on those that you selected. That is correct. So let me be more specific with my answer. That is correct if you save that selected, filtered, subsetted data set into a new variable. But it doesn't, you can see like I never reassigned tumor. I never said less than dash anywhere. And that's why I can keep saying tumor this, tumor that, and I get the original data set back. If I do, in the next example, I'll actually save the filtered data set. And then in that case, like when you call filtered, the filter data set, then the only values in there is what you have to work with. Let me show you what that looks like. Okay. So let's say we want to create another data set that's just a subset. Maybe we only care about the days where it's like 0, the first day and the last day of our tumor size values, right? So we can do our filtering, or selecting and filtering. So we can say tumor. Then we want to select on group, ID, day, and size. Maybe these are the columns I care about, and I don't care about the that other group with that tells me if it's a control or a study population. Then I want to filter such that day is equal to 0, pipe, or day is equal to 13, right? So we have this filter data set that is selecting columns and filtering our rows such that the day values are only 0 and 13. Let's say this is a data set that we like, we want to save and work with later on. Remember the assignment operator, when we assigned a loaded up data set into the variable called tumor, we can take this calculated data set and assign it to anything else that we want. So I can call this filtered and then less than dash tumor. And so now what this gives me is this variable called filtered. And anytime I call this is now that process data set that we were working with, but it's not affecting that first data set that we loaded up, right? And it's definitely not affecting that raw data set that we loaded from the CSV, right? So when we're trying to detach our raw data set with stuff that we're working with, using code is a good way to work with that, right? You don't want to be given like, here's my patient study, and then you are literally like line deleting data out of your data set, right? Like you might like save as the file and save as a filter, but then if I give you like the updated data set, you have to go through that deletion process again. Versus with code, I can say like, okay, I don't care how many times this tumor data set gets updated. I know I only want these filtered values, and I can get a process filter value through code. Right, so now I have this filtered value. And then the last thing I want to show you is we can take this filtered value and actually save it out to a CSV file. So in the tidyverse, there is a function called write_CSV. And then we can give it the data set we want written out, which is filtered. And then just like the path stuff that we saw, that I went over at the very beginning, we now give it like, where's the file path that you want this file saved to? So in our case, it would make sense to go into the data folder. So we can say data / and then I'll say tumor_filtered.csv and then run that. And then you'll see on the right-hand corner, or you can actually go onto your computer, go to desktop, go to this folder, go to the data folder, and you will actually see tumorfiltered.csv. That was a file that was saved to our computer, right? And you can, do I have Excel installed on this? Like, you can actually now open up this filter data set in Excel or whatever spreadsheet program you have. And this is the filter data set that's only values containing 0 and 13 with those columns. Right? So as far as like the first thing that we want to think about when we're thinking about building pipelines is, what is the input? What is the thing we want to do? And then what is its output? The input is going to be some file that we read in. It could be an Excel file someone gave you. It can be the output from another piece of code. we read in a file, we did some kind of transformation, and then we literally wrote out a file, right? And in terms of like managing expectations of what we're trying to get out from these 2 days is, if this is all you do is like, let's say you have a data set that's like any of those genomic data sets that have like hundreds of columns, and you only care about like 3 of them because those are like the SNPs that you care about. If all you do is load in that data set into R, filter out like those 3 SNPs with the ID, and then write it out so you can do your analysis without looking at the hundreds of columns, I would almost call that a win, because you'll, one, get practice on writing code in R, and then that is actually like the fundamental piece of building larger and larger pipelines, right? Because you're no longer modifying the original raw data set. You're not save asing the data set and then deleting things. So if you get an updated data set, you have to go through that process again. That's how a lot of those cases where you, give me a second, there's like a train passing by. So a lot of those cases where you are, those horror stories that you see where papers have to be retracted, it's because of like these Excel problems of like, oh, I forgot to update the data set, or like I forgot to like double click this formula to apply to all of my rows and only apply to like the first 50, and I got a new data set that like had 50 more values. Your code can prevent that from happening. Okay, so one last thing I want to very quickly go over, and we'll sort of touch upon this at the very end, but why does all of that stuff I talked about, spreadsheets, matter, right? So what we'll do is we'll actually start a data pipelining task. We'll go create a new file, and in general, you'll see that when I created a new file, my environment tab is still populated. I still have access to all of this stuff, this filtered data set, the tumor data set. In general, if you are thinking about creating data pipelines from your code, you want each file that we're working with to be independent on its own, right? And so one of the ways you do that in Rstudio is to clear out everything. So you are guaranteed that you are starting from a clean slate. Is you can go to session on the top and then click on restart R. And the keyboard shortcut is Control-Shift-F10. And what this will do is you'll notice that the environment panel is now going to be empty, and it's going to restart the R session. And if I do something like read_CSV now, it's going to say I can't find read_CSV, right? It completely undid all of the libraries we loaded, undid all of the variables we created, like filtered is no longer going to be found because it doesn't exist. And this is great when we start a new script, because when we're thinking about pipelines, that next piece in the pipeline is just going to run that thing from top to bottom. And so it's not going to have access to all of the libraries and variables and files. And so this is one way when you're thinking about building pipelines, to essentially hit Control-Shift-F10 or going to session restart R, to make sure you're always starting from a clean slate. And this makes it so your code, I can literally give any one of you this script, and you can run it on your computer, assuming it's in the right location. And I don't have to, and you don't have to worry about like, did Dan create like some variable that's in here that I don't know about, et cetera, right? That's the thing you don't want to. You don't want code from one, you don't want things from one file contaminating things in another file. And so here we have to load up tidyverse again. So we're going to say library tidyverse. And then we can say filtered gets read_CSV, and then data slash, what did I call it? Tumor filter.csv, right? And so we've actually just created a pipeline. We took the output from one file and it became the input to another file. To round off this pipelining task, we can hit Control-S to save. And then I can just literally name it 02, and then we can call it descriptives, because I'm going to show you how you can run like very basic descriptive statistics off of this. Okay, so we have this filtered data set. So we can either type filtered or click on this thing to look at it. And so let's say I want the average tumor size of the first day for everyone. How do I do this? So the first thing was I want the, forget the fact that I said average. I want what, I'm taking an average of what? It is the values where day is 0, because I said like the first day, right? So let's do that part first. How do we filter our rows for the first day? We use the filter function. Filter where the values of day is double equal to 0 or equal 0. And now we have all of the data sets, all the values where day is 0. So now what was the next task? Okay, I have all of the rows. Now I want to have the columns. So we can say select size. And so now I have all of the values where day is 0 and just the size column. And then R has a built-in function called mean. This will work. R has a function called mean. And this isn't going to work right now because mean needs essentially what R calls a vector of numbers. So think of like a comma separated list of numbers. A comma separated list of numbers is actually different from a column of numbers, which is like an Excel column, right? So if we take back the last step where I went to say select, you can see that this gave me size as a column. But if I want the values of size as just their individual values, there is a function called pull. So I'm pulling the size column. So instead of selecting the size column, I am pulling the size column. And you can see the output is now different. So I will paste it so you will see the difference. So select gives us the column, and pull gives us the actual column values, right? So you can see that they're printed out differently. Now that we have just the raw values, we can then say mean, and we can calculate the mean of all of the values where the mean size for day is 0 from our filter data set. And we can go even further. So I'm going to copy this whole thing and I will paste this into chat. So that's what I typed in. And we can build on this even more, right? So when we're trying to figure out what's going on in our data set, let's say I want to filter where the group is equal to 1 and the day is 0. You can now see that the overall mean is 51.6 essentially, but the group 1 mean is 55.6, right? It's a little bit bigger. We'll cover statistic stuff on Thursday for a very little bit, but this is how you can explore your data set to find different groups. But now if my question to you is, I want the average size for day 1 across all of my groups. Now you run into this situation where like, okay, I am gonna copy this, right? So, and then I'm gonna replace group to 2. And so that's my average for 2, hit paste, replace this with 3, and I'm just gonna keep going until like I end up with a value that doesn't exist, right? Like 5 doesn't exist, so I end up with nothing, right? So that's one way you can do it. And I'm not going to say it's wrong, especially if you're learning. Like, if this is what it takes to get your work done, like, please go do that. But there is a better way, right? And the way we can set that up is if our data set was, this always comes back to like that spreadsheet chapter. If your columns only store 1 bit of information, a lot of this data processing can become really, it can look like magically like simple in some way because it can do so much with a few lines of code versus like you trying to struggle with this in a spreadsheet program yourself. And so what do I mean? My question was, okay, I want the average size for the day 1 across all of my groups, right? So yes, the question was, can I get parameters like mean and median? So you can also say median, and I'll give you the yeah, median. Short answer is yes. There's a way to do median. Actually, why didn't this? Oh, that's because 5 doesn't exist. Okay, that makes sense. Yeah, so there is a function called median, and you can get the median as well. But there's actually a nice way to do all of those calculations in one go, which is what I'm going to show you before we finish off today's class. And this is known as a group by operation. So think of the question that we're asking is average size for day 0 across all groups, right? So if you want, you can put a little hash and it'll be a comment. So this is code that will exist in your file, but not be executed if you try to run it. It's a good way to take notes inside of your code. So this is the task that I want. So we want, at the end of the day, we want some value for average size, but we can filter our data set for day is equal to 0. So let's do that first. So we can say filtered is going to be filter day is equal to 0, right? So now I have part of my data set. It's the values where day is 0. The next thing is I want some calculation, which is mean, but for all of my groups, right? And I don't want to have to manually specify that. And the trick to this is in the tidyverse, there is a function called group_by. And in here, you say, you give it the grouping variable. So right now I can say, I care about the group column. And right now it doesn't do anything. All it says is like, there's groups and there's 4 groups, great. But now R understands that if I were to perform some calculation, it knows to do it for each group one at a time, right? So it's essentially gonna subset on group 1, do a calculation, subset on group 2, do a calculation, subset on group 3, do a calculation, right? The way group bys are handled, there is this notion called split, apply, and combine. So group by is going to split our data set into 4 different parts. It's going to apply a function, in which case it's going to be the mean function, and then combine it all together to give us one data set at the very end. So that's what group by is doing. It's called split, apply, and a combine. And the way mean functions work is the mean as a function is a function that can take a bunch of numbers, but the end result is always one value, right? So that is called a summary function. And so we can say, I want this group by operation to perform a bunch of summary functions. So we can say pipe into summarize. And then I'm going to work out this example and hopefully it makes more sense when you see the results. I want a column called average size, and it's going to be equal to the mean of the size column. And so that's a lot of stuff on the last line, but if we run the code, hopefully it makes a lot more sense. When we run this, we get a data set back. One column is called group, and we have 1, 2, 3, 4. And then the other column is called average size, and it's the average for that group. It's the average size value for that group. And so we've essentially created our first summary statistics table. And instead of like in above, what we did was filtered on group equals 1, filter on group equals 2, 3, 4, et cetera, et cetera. We actually did the same thing with this one line that said it's group by on the group, and then pass in the summary statistic that we want. Summarize is a function of tidyverse, yes. One thing that you can, that'll help you if you're trying to look for help, if you put your cursor between the end of the function and the beginning of the round bracket, and you hit the F1 command on the right-hand side, it'll open up the help page. And so technically summarize is under a function called dplyr, but dplyr is under tidyverse. And then you can get all of the, how do you use the summary function or summarize function. And so the question I got before is like, okay, what about min, median, max? So now I can say min size is equal to min of size. And just to make the code a little bit nicer, I can put a new line across each feature. I can say max size is equal to max of size. And then median size is median size. So now I can create all of these summary statistics, but it knows to handle for each value of group. So if you just run summarize on its own, it's not going to do anything, right? Because you actually didn't give it a summary function. You need to, so this was a lot of stuff I typed. You need to actually provide it a summary function. So what is an example of a summary function? Is the mean, the min, max, median. You can actually pull out a very specific quartile. So if you want like the 68th percentile, you can actually get that as well. Right. So the key trick here was instead of copy and pasting your code, like doing some summary statistic for each group, you can pass it into the function called group by, and it will now know anytime you do a calculation, or it will break up your parts by that, what you specified in the group. In Excel, there is an actual feature that does this as well. I think it's literally called group by as well. This is just this way of handling summary statistics is pretty common across all data programs. And so this will prevent you from, making copy paste error appear, right? Because it's very easy to miss, like maybe this is still 1 and I think it's the value 2 and I have 2 values that are like slightly different. I think they are the same averages when it's in fact like a copy paste mistake. But having the group by summary actually prevents that from happening. Okay, so that is actually everything I wanted to cover for today. In your emails that you got yesterday, there was a link to the post-workshop survey. It is also on Etherpad, which is like below me somewhere. It's bit.ly/ds4biomed-etherpad. So if you go there on line 14 of the Etherpad, there is on line 19 of the Etherpad, there is a link to the post-workshop survey. You can choose to give feedback without being part of the study, or be part of the study and give feedback. It's literally just so I know what to do for Thursday and make this better. Also, just as the last take, if you want an actual question to work on, for now, I will end up with this as the last question, and I'll post the solution on the GitHub page with all of the code that I have. So if there's something that you missed on the code that I typed, I will, right after today, there will be a follow-up email where you can literally see all of the stuff that I typed. And I'll also put a solution to this exercise as well. So we'll give everyone back roughly 8 minutes of their time. If you have any questions, just do let me know. If you can right now, spend the next 8 minutes working on the post-workshop survey. That would help a lot. The people who took the persona survey in the summer, thank you for that. It actually made this, it helped focus the content for this material. So it does play a big role into all of this. But if you have any general questions, just post them in chat. Essentially, what we are going to talk about on Thursday is pivoting tables. So if you have a data set that makes a lot of sense for data entry, but doesn't work very well for like this type of data processing, how do we transform our data from one shape to another? And then I will do a very quick introduction to plotting and fitting a logistic regression, which is probably in medicine the most common model that's going to be used. So that is the goal for Thursday.
We thank you guys for all being here. We'll stick around for a few minutes. If you have additional questions, please check out all our materials. We hope you guys got something out of this, especially with how to organize data in ways that will make your analysis life much easier. We are happy to help as well with other data projects you might have, so please don't hesitate to reach out to us in data services, and we can consult with you in a variety of ways to help you with your data questions. And we look forward to seeing you on Thursday as well.
Yes, and there will be a totally separate calendar invite going out somewhere between today and tomorrow. I just didn't want to confuse people with a bunch of calendar invites. So we will have a calendar invite with the links to Thursday's class as well.